doing a project on bioengineering. So what is bioengineering? Bioengineering is basically changing the characteristics of an organism by manipulating its genetic material. So how far is too far? With the current advances in bioengineering today, we can manipulate our foods, our own bodies, and maybe even one day possibly clone ourselves. And so with that, that's what we're going to talk about in our presentation today.
To start off, we're going to talk about genetically modifying organisms, or GMO. GMO are plants that have been genetically engineered with DNA from other plants. What is GMO? How does it work? When scientists genetically modify a fruit, what they do is actually a very simple process. They first find a trait that they wish to put into the fruit. For example, seedless. Seedless is a very common one. Many people would rather have a seedless watermelon than a normal watermelon. So once they isolate that gene with the desired characteristic, they physically insert that gene into a separate seed using plant tissue. How common are GMOs? GMO is extremely common in America. Scientists started genetically modifying crops in 1982, so that's over 30 years ago, or 20 years ago, when the FDA approved it for American farms in 82. Today, 93% of our soy and cotton plants are GMO, and about 86% of our corn is GMO. These rates are still rapidly growing every day here in the US. But for 40 other countries and worldwide, GMOs have been significantly restricted or banned due to the uncertainty of its safety.
The proponents of GMO emphasize the potential benefits increasing agricultural production. GMO has been part of our food for the past 30 years. And the primary benefits of GMO foods is better overall quality and taste, which through the modification of foods, the flavor can be enhanced, which like peppers can become spicier, corn can become sweeter, or difficult flavor can be palatable.
But is taste really worth the health? Research shows that inserting foreign genes or toxins into other plants causes the plants to try and fight off that external allergen. Just like our body fights off germs when we get a cold, this causes the crops to create toxins that could be harmful to the animals or the humans that eat those specific fruit or vegetables.
Another primary benefits of GMO food is more resistant to disease. Plants and animals that have been genetically modified can become more resistant to unexpected problem of disease. Think of it as a vaccine for that plant or animal, except for that vaccine encoded in genetics instead of the shot given to immune system.
So maybe that plant is stronger and more resistant, but what causes that resistance to pesticides is crossing fragile plants with sturdy plants. When scientists did this to a certain strain of corn in Iowa State, they changed the genetic makeup of the pollen in the corn, and it greatly affected monarch butterflies in that area. Monarch butterflies were not used to the pollen of the milkweed plant, which was the plant that the corn was crossed with, and it caused a 58% decrease in monarch population. Monarch butterflies may sound like a very fragile animal, but if it's decreasing the population 58%, think of what it could do to humans.
Another primary benefits of GMO food is more nutrition benefits. GMO foods can have vitamins or minerals added to them through the genetics modification to provide greater nutritive benefits to those who eat them.
This is true, that crops could theoretically could have more nutrients, but is that really what large produce companies like Monsanto are actually interested in? No, because in 2003, the American soybean was so altered that it caused it to completely lose the quality phytoestrogen. All the compounds of phytoestrogen was gone from those soybeans. That same nutrient is believed to fight cancer and protect against heart disease, which is the main reason people eat soybeans. So we're not truly using the technology for good, or at least large companies aren't. Bioengineering is essentially the alteration of life. So if it is ethical to alter the genetic information of plants, is it ethical to alter the genes of humans or animals?
That's a very good question. Like Jen and Katie said, we've been using and changing plants for at least 30 years. We've been doing the same thing with animals for at least that long, if not longer, in lab studies and whatnot. But it's changed greatly in '98 when they discovered stem cells. Stem cells, I could tell you all day what they are, but this will give you a very clear picture of what they are.
Hey everybody, just a quick heads up. Now, imagine this is a stem cell.
Stem cells are unique because they have the ability to generate new cells of almost any kind. They all start off as unspecialized cells, but
Given the right chemical and genetic signals, the stem cells can divide to form slightly more specialized cells of different size, shape, and function. And after a few more cycles of division, these can give rise to highly specialized cells, like heart muscle cells, for example, that help your heart pump the blood around your body. Given a different set of signals, this same unspecialized stem cell can go down an alternative pathway and give rise to a different type of specialized cell, like a neuron that transmits electrical signals in the brain.
compared with stem cells from embryos, adult stem cells give rise to a smaller number of cell types, usually those of the organ or tissue in which they're found.
Now, researchers have found adult stem cells in more tissues than previously thought. Bone marrow, skin, brain, liver, eyes, and this has led to research into using the patient's own adult stem cells to repair damaged organs.
And that's exactly the focus of the trial Michael Taylor's taken part in. Led by Professor Anthony Mather at the London Chest Hospital, the trial is vitally important because heart disease is still on the rise.
Cardiovascular disease is the #1 killer in the UK, with nearly 3 million people suffering with heart conditions in the UK, and about 800,000 people suffering with this condition of heart failure. He's got a condition which makes the heart very weak and baggy, and it doesn't work very well as a pump. And what we're trying to do is see whether his own stem cells can actually repair that heart and make it pump effectively again.
So if you're looking to repair heart tissue, do you need to use heart stem cells?
Ideally, yes. The problem is that there are very few of those cells in the heart, and we've only recently discovered that they're there. And the few heart stem cells that are present clearly aren't capable of repairing it. And that's really where we step in with our trials. By using bone marrow as a source of stem cells, we try and enhance the stem cells in the heart and their ability to repair the damage that's been caused.
This trial tackles heart disease on two fronts. Michael's bone marrow has already been stimulated to release enormous numbers of stem cells into his blood. And now, more are to be harvested directly from the bone marrow in his hip.
You okay?
Yeah, fine, thank you. Yeah, it wasn't that bad at all.
Good.
As this is still a trial, only some patients will have these stem cells re-injected. Although Michael will get an injection directly into his heart, neither he nor Professor Mather know if it will contain his stem cells or a placebo.
We're about to put either stem cells or placebo into his coronary arteries. We won't know which until the end of the study when we've treated all of our patients.
This uncertainty helps the researchers to rule out the placebo effect from any positive results they achieve. Professor Mather feeds a tube through Michael's blood vessels right to the site of the disease in his heart. Incredibly, Michael remains fully awake throughout the entire procedure.
All right, where's that?
It's fine, thank you.
You can't feel anything?
No, not doing
So that's the first artery done, now the second one, and then one more, and then we're all finished.
That procedure was showing his own adult or stem cells, but there's also embryonic stem cells that she mentioned. There's a big difference in like how people look at the stem cells. Where embryonics, we'll let him explain.
Yeah. As you heard in the video, we all have stem cells. They're in our body each and every day. But how do we activate them? They don't just magically grow into other cells that go specialized and become hard tissue. They have to be like stimulated in the right scenario so that they can become that. And embryonic stem cells are like a week-old embryo from a fertilized egg. And unlike the adult stem cells, the embryonic stem cells, they have a large and much higher potential to become any other type of specialized cells that can help our body and possibly cure multiple diseases that we have ongoing in this world. And as you can see in the picture, it can become just about anything. And embryonic stem cells are more useful. And also, as a mental note, Barack Obama also allowed the use of embryonic stem cell research. Unlike Bush, he denied it completely, and Obama put into effect and is funding stem cell research for Americans to possibly cures for many diseases.
Some of those cures start with the adult stem cell transplantation, which treats diseases. And what they do with the blood transfer is they take blood that they've cultivated in a lab from your body that has an increased number of stem cells and then put it back into your blood. And what it does is it helps fight diseases in the blood and immunity, such as sickle cell anemia, and it helps certain cancers like breast cancer, and like other diseases that like affect the blood in particular.
Pro-life was a big deal when it comes to embryonic stem cells because it is a fertilized egg. Although it's only like a week-old embryo, it still has the potential to become life. And that's why under the Bush administration, he completely disagreed with the idea and didn't put it into effect. So that could be a potential con for using embryonic stem cells.
One pro of using stem cells is with like the tissue grafting. What I say right here with Russell Turnbull. This man, what happened to him? He was like I can't remember if it was a bus or a train per se, but he was in public transportation and a fight broke out. And during that fight, he tried to stop it. Trying to help out. He had ammonia sprayed in his eye, made him blind in that eye. So for 12, 15 years, he went completely blind in that eye until he volunteered for the research. And by taking stem cells from the good eye, cultivating it in a lab for 2 weeks, and then replacing it into the bad eye, over time, he now has almost full vision again in that bad eye. So there's one benefit.
Another big problem is when it comes to people's real personal thought and ethos and ethics, because it's certainly continuing to be researched and its full potential has not been tapped or developed yet. So a lot of people, they like to criticize something that has not been fully developed, and that's a negative aspect.
One of the positive aspects of it, though, is with the development of new medicines and also new treatments for basic things. Like we don't even have a cure for Ebola or anything. They're developing things right now by looking at stem cells for it. But with the new medicines, instead of using animals to do lab practices, they put the drugs directly onto the stem cells. That they wanted to cure liver, they used liver stem cells, and see how it affects the human cell directly instead of using rats. So that's a plus.
Another negative aspect, because it's underdeveloped, a small percentage of patients who have taken stem cells have later developed possible signs of cancer, and that could be a problem. But although it's something that we just started and we're still working the bugs out, that can be also a con.
Well, this here is a list of just a short list of the current uses for adult stem cells. It's used in the treatment of so many different diseases right now, including Hodgkin's, Parkinson's, different cancers. Leukemia is being treated using stem cell research. and there is just a very large variety of different diseases are being treated right now using this. I focus mostly on the adult stem cells because that's the ones we've been allowed to like mess with for the longest amount of time. And embryonics is still very new because of all the different debates about the ethics of it. So, but because of this, many diseases are being treated and being helped, and hopefully the potential for that could be limitless.
So what does this lead to? If we can grow our own tissue and organs and put it back into ourselves, why can't we just replicate a whole nother body?
Well, that's really controversial, but it is a good question. And so, cloning. What is cloning? Cloning can happen in nature, and it does very often. Like you see with identical twins, they have the exact same genes. They're perfect genetic copies, but if we're speaking artificially, there are two types, which you see here: artificial embryo twinning and somatic cell nuclear transfer. And the artificial embryo twinning, I'm not sure whether it's more common, but it's more similar to what happens like in the womb with identical twins, because basically all that happens is an embryo is separated in a petri dish into individual cells, and after some time it's placed into a surrogate mother, and that's where it can develop and a baby's born. But the more controversial is the SCNT, which here there is an egg donor. The nucleus of the egg is removed, and a cell donor, a somatic cell, which is any cell besides the two sex cells, is enucleated through electric fusion, and then the egg is fused with the cell, which forms an embryo, and then a fetus is developed, and there's a surrogate mother with a cloned baby. So back to this: is cloning an organism the same as cloning a gene? Well, it's a little bit different, because a gene is just one gene out of an entire organism, and in a lab it's definitely easier to see the development, and if something were to go wrong with a single gene instead of a whole person or animal. So, pros and cons.
Well, I mean, if you just think about cloning, I mean, what is cloning? I mean, you're basically recreating the entire human body from scratch. Well, not necessarily from scratch, but from other cells. So think about it. What if you have someone who's an amputee, either missing a leg or missing an arm? Well, if you simply clone a human being, then you're going to have an arm or a leg to transplant to that person. Now, when I say clone, I don't mean necessarily cloning like an entire functioning human being. All I'm saying is that if we were able to clone a human being, but just without the brain, so they're not able to feel pain, then it would be a very viable source of limbs or organs. Because right now, there are people on organ transplant lists waiting years to get their organs, and many of them don't make it until there's an organ available. But if we were able to clone people, and just without the actual brain, so they're not able to feel pain, they're just a body, but without the brain, then we'd be able to use those organs and those limbs to transplant to people who actually need them.
A con is its extremely high failure rate. With somatic cell nuclear transfer, only 1 in 3%, or 0.1 to 3%, which is 1 to 30 in 1,000, are successful. Which you could also look at it as though 970 to 999 in 1,000 were failures. So that isn't very good. But
Well, I mean, like, in response to the fact that you're saying it has a high failure rate, I mean, sure, it has a high failure rate, but you're not really losing that much from failing. You lose a few cells, you lose a bit of money, and you lose a bit of time. But once you finally get that success, then you're going to be able to save or drastically improve dozens of lives just from a single body. And also, with cloning, you're able to eliminate defective genes. Say you have a couple and they want to have a child together, but unfortunately one member of the couple has some kind of genetic disease, like Hutchinson's disease or Addison's disease, and they don't want to pass that disease on to their children. Well, what you can do is you can simply clone the genes of the other person in the couple, so they can still have a child, but they don't have the same genetic defect. Because when you're cloning a person, it's not necessarily creating a person that's entirely similar. When you're putting the genetic material into the nucleus of the somatic cell before it turns into a fetus, you're able to take out some of the genes which could negatively impact someone's quality of life.
There's also, though, health problems in later stages of development, and even before later development. An egg that's had the nucleus removed, the transfer couldn't be compatible with the nucleus of the somatic cell, and it could just fail entirely that way, or it couldn't divide or develop properly, and that would fail, or implantation of an embryo into surrogate mother might fail, or the pregnancy itself might fail. And there's also when an animal or person is born, there's LOS, large offspring syndrome, which can lead to abnormally sized organs, which can lead to breathing or blood flow problems. And if they don't have LOS, then they're still susceptible to kidney or brain malformations, or impaired immune systems.
Well, I mean, that's a very valid point, but the thing is, like if someone is having, say, a heart attack and you need to have like an artery opened up or a stent put in, sure, there are risks with every surgery that we do, but we still do the surgery because statistically, the person's going to be better off having the surgery, taking the risks of infection or blood loss, than now having the surgery done. So with cloning, it seems very worthwhile to take these risks because even if you have just one success out of 1,000, you're still saving dozens of lives. And in a similar sort of thread, cloning gives us the ability to save endangered species. You have some species of animals where there are only 40 or 50 living members alive, and that's just not enough in some cases to sustain the population. With cloning, you can clone the healthiest members of that species, and you can keep that species going. So we could prevent we could increase the biodiversity we have, and we could prevent a lot of species from going extinct.
But a transferred nucleus doesn't have the same programming as a natural embryo. So what scientists have to do is they have to fully reprogram a nucleus, otherwise the embryo can develop abnormally or fail. And that is a waste of time and money, and seeing how often it fails anyway, it's definitely not a good thing.
Well, abnormal gene expression does occur a lot of times when you're creating a clone, but we also see failures with things like stem cells and other kinds of technology where you're trying to fundamentally change how an organism is functioning. But this is something that is really just needs time to be worked out. And if there was funding available for cloning, and if there weren't any laws preventing cloning, then researchers would have the time and the resources and support that they need to actually go in and find where the flaws are and fix them before it actually becomes a more mainstream technology.
Something that I think even pro-cloning people could agree with is that telomeres are in a DNA sequence. At both ends of a chromosome, there are telomeres, and through cloning, they shrink every time DNA is copied. But that happens naturally too. Like every time your DNA replicates itself, your telomeres get shorter. But through cloning, some clones have abnormally short telomeres at birth, so their cells age faster than normal people, or animals, so they wouldn't live as long of a life. Probably, if it were to happen, it'd be extremely short and unsuccessful.
Well, see, it is true that with the technology we have so far, a lot of times when we create a clone, that its telomeres are shorter than they should be. For example, Dolly the sheep, the first mammal that was cloned, it only lived to about half or about roughly 60% of what you would expect the sheep to live to given the circumstances it was in. But with cloning, we also have the unique ability to not just recreate a cell, but also to alter the telomeres. So theoretically, with more research, it would be possible to actually lengthen these telomeres and basically create human beings who can live much longer than average. Because when telomeres are longer than average, then a person has less risks has a lesser risk of developing things like cancer or autoimmune diseases. So this is something that can definitely be researched and it could be fixed.
These are very good points.
All right. So with just looking at bioengineering as a science, I mean GMOs and stem cells and cloning, I mean we have to ask ourselves where do we draw the line? I mean most of us are okay with GMOs. I mean I'm guessing that every single person in this room has eaten at least a few foods in their lives that have GMOs in them. But I mean like once you sort of go from GMOs to stem cells, that's where a lot of people they don't want anything to do with stem cells because they think it's unethical or they think that we're trying to play God. And the amount of supporters of bioengineering decreases even further when you get to topics like cloning, where the vast majority of people don't agree with cloning. They think that it's not a good thing because they think that as humans we're sort of overreaching our boundary. We're doing things we're not supposed to do. But I mean, other people would say that humans are unique when compared to other animals because we always try to do the things that we haven't done before. We try to go further than we've ever been. So it's definitely two sides of the argument you have to consider when you choose what to support. And I mean, another argument that kind of ties in the first one is, does unnatural equal bad? I mean, sure, people say that GMOs are bad because they're artificial and all that, but are they bad just because they're artificial? I mean, the electricity we use, the phones we use, the cars we drive, they're all man-made, but just because they're man-made doesn't mean they're bad. I mean, our lives are vastly great, we have a vastly greater quality of life than our caveman ancestors because we've used technology to sort of further our way of life. And the really third argument is there a right choice? I mean, how do we know if cloning or stem cell research is going to be a good thing until we go and try it? So it's up to you guys to decide if we as humanity should take that risk to go ahead and research these technologies, even if there's a chance that problems can occur and that this technology can be abused. So you have all the information. The choice is yours.